Hello, welcome to this masterclass on how to identify variants and profile methylation in human genome samples. My name is Tomek, and I am a member of the technical training team at Oxford Nanopore Technologies. This masterclass is part of a series that takes you through the whole Oxford Nanopore sequencing workflow, detailing every step from experimental planning to data analysis. Now, this masterclass is a little special. Instead of focusing on a particular step of the sample to answer workflow, we will discuss how we can apply best practice from each of the other masterclasses to identify variants and methylation profiles across the human genome, following our recommended end-to-end human variation workflow. We will start by exploring why Oxford Nanopore sequencing is particularly suitable for multiomic analysis of human genomic samples. Afterwards, we will go through every step of the human variation workflow, starting from sample preparation and going all the way to generating analysis results. At the end, I will provide you with online resources for your further exploration. So let's get started with a brief introduction in which we'll explore why Oxford Nanopore sequencing is so fantastic for analyzing human genomes. Taking a step back, at the heart of biology lies the central dogma. DNA stores the genetic blueprint. RNA transcribes this information, and proteins carry out the functions of life. To truly understand complex biological systems or uncover the mechanisms of disease, we need tools that can analyze all three layers, DNA, RNA, and proteins, together. But here's the problem. Legacy multiomic technologies can be complex and cost prohibitive. In genomics, traditional methods like sequencing by synthesis, microarrays, and bisulfite sequencing are often expensive, require complicated workflows, and still fail to capture critical details like structural variants or all methylation patterns. Similarly, in transcriptomics, techniques like RNA-seq are piecemeal solutions that require multiple steps to reconstruct full-length transcripts or RNA modifications. Oxford Nanopore aims to redefine this approach to deliver ultra-rich multiomic data from a single platform. So how do we simplify the process of generating rich, comprehensive biological data? Well, let's focus on analyzing genomic DNA. With traditional sequencing technologies, answering complex biological questions often requires juggling multiple steps. You need to prepare several libraries, use different sequencing platforms, and then navigate complex data integration from multiple analyses. This approach is time-consuming, costly, and introduces potential for errors or missed insights. Oxford Nanopore eliminates this complexity. With just one DNA sequencing library, one sequencing run, and a single streamlined analysis workflow, you can generate ultra-rich data. This includes everything from structural variation and single nucleotide variants to methylation and phasing, all in one platform. Oxford Nanopore sequencing isn't just simplifying workflows. It's actually unlocking a level of detail and integration that legacy technologies can't match. Let's take a closer look at how this efficiency translates into meaningful results for biological research. In the context of genomic studies, traditional platforms are capable of resolving single nucleotide variants and indels, which can provide insights into some of the known human diseases. However, Oxford Nanopore sequencing can deliver everything traditional short-read sequencing technologies can, and all this enriched with new insights not previously possible. Small variants, including SNVs and indels, can be seen across more of the genome than ever before. Long reads mean these can be accurately phased, and native sequencing allows you to assess the biology that's actually there, with base modifications included. Additional benefits include capturing complex structural variants and copy number variation. A lot of these variants are relevant in the context of studying human disease, and you can gain all these insights from a single assay with flexibility and automation. From a single PromethION flow cell, it is possible to investigate the human genome at around 30x coverage and profile high-quality genomic and epigenetic variants. Our latest metrics, shown here and obtained with high-accuracy base calling, include methylation detection at single base level at 99.7%. That's more accurate than by sulfide sequencing. Structure variant calling over 96%. And as the benchmarking is actually limited by the truth set, this is as good as it can get. Megabase scale phasing and 99.7% SNV F1 scores across the whole genome. To enable access to these insights in human genomes, our teams have worked hard to develop end-to-end protocols covering all the steps from sample to answer. Detecting variants and methylation in human samples is not the only end-to-end workflow we have developed in the space of human genomics. And this slide reviews their key features. Starting with the human variation workflow, it allows simultaneous analysis of SNVs, SVs, CNVs, STRs, and methylation. With a sample to sequencing time of 4 to 6 hours, depending on the sample type, and automated reporting, this workflow provides a seamless and efficient solution for studying genomic and epigenomic variations. Secondly, when working with liquid biopsy research samples, the cell-free DNA workflow is ideal. It supports both short and long cfDNA sequencing while retaining methylation data, providing insights into tissue of origin, and epigenetic profiling within just 6 hours. For oncology research, the tumor-normal workflow excels in retaining all methylation markers alongside genetic variants in tumor-normal research samples, improving the classification accuracy of tumor types and the identification of driver mutations. Lastly, the single-cell workflow enables full-length transcript sequencing for isoform reconstruction, gene fusions, and SNP detection. It generates around 80 million cell-assigned reads per flow cell and supports 10x genomics technologies, including Visium spatial mapping. Collectively, these workflows offer flexibility and scalability, ensuring no critical data are missed, and providing researchers with a clear path from starting sample to comprehensive insights. Today, our focus will be the end-to-end human variation workflow. And in the next section of this masterclass, we will explore this workflow in detail. Our end-to-end workflow for studying human genomic variants covers preparation, sequencing, and data analysis, giving our users control at every stage. We'll look at each of these steps in a moment, but for now, here's an overview. For library prep, we recommend the ligation sequencing kit, which is also available in an XL version and is compatible with automated solutions. For most efficient generation of human genomic data, we recommend using our PromethION flow cells and sequencing devices. Finally, for downstream analysis of the data, we provide our Epitome human variation workflow, which is also compatible with further tertiary annotation platforms. In the next sections, we will talk more about each of these stages in the context of analyzing human genomes. To learn more about sample and library preparation, sequencing devices, or data analysis in a broader context, please refer to the dedicated masterclasses in this series. Well, let's get started with how we recommend preparing research human samples to study variation and methylation. As presented in the masterclass about DNA and RNA extraction, choosing the right extraction method is essential for a successful sequencing experiment. Here is a table summarizing our experience of extracting human genomic DNA from different research sample types. For each of these, our teams have tested available extraction methods and selected those that perform best. In the table, you can see the required amounts of starting material and expected DNA yields. For most samples, the extraction method can be automated. For whole genome sequencing and variation profiling, we find that blood and saliva samples perform the most robustly. And so in today's masterclass, we will go through the end-to-end workflow, starting from blood samples. For other sample types, you can find the extraction protocols in the documentation space on our website, including complete end-to-end protocols for saliva and buccal swabs. Starting with extraction. To isolate the DNA, we need at least 1 milliliter of human blood and the QIAGEN PureGene blood kit. After the extraction, we recommend quantifying the extracted DNA using a qubit fluorometer and assessing its fragment length distribution using Agilent femtopulse system or equivalent. 1 milliliter of blood should yield between 10 to 30 micrograms of DNA, with fragments longer than 50 kb present in the sample. While size selection and fragmentation are optional steps in our standard ligation sequencing protocols, our end-to-end workflows include instructions on these steps to optimize the sequencing performance and read length. Therefore, we recommend first treating the isolated DNA with our short fragment eliminator kit to deplete fragments shorter than 25 kb. Afterwards, light shearing with the Megaraptor device from Diagenode generates fragments with lengths centered around 30 kb. Besides the initial assessment of the extracted sample, our end-to-end protocol advises on how to perform quality check at several points through your sample preparation, including quantification with a qubit and read length QC, and checking for potential chemical contaminants. This will help make sure that your sample will give high outputs of long reads in sequencing. After the size selection and fragmentation, we take 3 micrograms of this prepared DNA into the ligation sequencing kit library preparation protocol. You can learn more about the benefits of the ligation sequencing kit in the library prep masterclass, but here we choose this kit for the highest level of control over read length and output. The library prep process begins with enzymatic repair of nicks in the DNA and the generation of uniform ends with 5' phosphates and 3' adenine overhangs in preparation for the next step. And next comes the attachment of sequencing adapters to the ends of the molecules using a ligation-based method. These sequencing adapters feature a pre-bound motor enzyme, which, as you will have seen in other masterclasses in the series, unzips the double-stranded DNA molecule and controls its steady translocation through the nanopore for sequencing. After a cleanup step, the sample is then eluted in elution buffer. For the human blood end-to-end workflow, we increase the volume of elution buffer in this final step. This is so we can load our library 3 times, once at the beginning of the sequencing run, and twice more after washing the flow cell. And we'll go into this step in more detail in a moment. We expect between 1200 to 1800 nanograms of final library. The ligation sequencing kit protocol is completely PCR-free, preserving high molecular weight fragments of DNA and native modifications for direct sequencing, and ensuring that regions of the genome that are challenging to amplify are captured. While our end-to-end workflow describes the library preparation in a manual fashion, at Oxford Nanopore, we have partnered with multiple automation vendors to support the automation needs of our users. These partnerships enable support at every level of throughput, from standalone systems within research groups to high-throughput integrated systems for population-scale sequencing projects. In particular, an automated ligation sequencing kit protocol has been developed on several available platforms, opening the possibility of scaling up projects aimed at detecting variants and methylation in human samples. We're now going to move on to the next step of the workflow, sequencing. As explained in our PromethION flow cell loading masterclass, our platform can be scaled depending on the project needs. And the first layer of scale lies within the flow cells. While MinION flow cells are best suited for smaller genomes or low-pass sequencing of large genomes, we recommend PromethION flow cells for profiling a whole human genome. This is due to their capacity to efficiently generate high outputs of sequencing data for good depth of coverage across the human genome. And this is why the PromethION family of Oxford Nanopore sequencing devices is best suited for human genome projects. Both of the devices shown in this slide, the PromethION 2 integrated in the top left and PromethION 24 at the bottom, allow independent control of the PromethION flow cells and include high-end GPUs for data processing in real time. They also offer the possibility to run the downstream analysis directly on the device. The major difference in choosing between them comes down to the number of flow cells you need to run. And this is where the second layer of scalability comes in. Let's look at each of these devices in more detail. The PromethION 2 integrated device enables sequencing on up to 2 independent flow cells and can run around 200 flow cells per year, making it ideal for labs looking for PromethION scale sequencing for smaller sample throughput requirements. Scaling up, the PromethION 24 enables sequencing on up to 24 flow cells at once, producing a capacity that reaches around 2,500 flow cells per year, making it the best choice for high throughput on-demand sequencing. When talking about sequencing human genomes, we must mention flow cell washing as a key step to optimize the performance of every sample. In this process, the flow cell wash kit, or its XL version, is used to wash the flow cell. After this, another aliquot of the prepared sequencing library is added to the flow cell, and sequencing continues. This wash step helps to recover unavailable pores on the flow cell and increases overall output. As you can see in the graph, we have tested the impact of flow cell washing on libraries with different fragment sizes. Shorter human DNA libraries extracted with other methods do not require washes to generate sufficient amounts of data. As you can see for the dark blue line, which shows data from a run in which the human genome is sequenced in 10 kb fragments. However, the rate of data generation from libraries prepared from blood samples, as described in our end-to-end protocol, tends to plateau due to the reduction in pore availability. Flow cell washing and reloading can significantly boost the sequencing output from such libraries containing long and ultra-long fragments of DNA, as you can see from the light blue line in the graph. By resetting the number of active pores, as demonstrated in the pore scan graph below, this approach increases the data generation rate after each wash, enhancing overall sequencing capacity and efficiency. And this is why we recommend at least one wash and reload for DNA libraries with fragments above 15 kb, and 2 reloads when sequencing our 30 kb libraries prepared with the blood sample end-to-end workflow. The washing itself takes approximately 1 hour and 15 minutes, and we recommend setting the total sequencing time to 100 hours. However, please remember that our flexible platform allows you to stop the sequencing run or start the analysis as soon as a sufficient amount of data has been generated. And that brings us to the final step of the workflow, data analysis. Here, we will explore how to use 2 pieces of our software, Minnow and Epitome, to generate the final results. You can learn more about Minnow and Epitome in the dedicated data analysis masterclass. But as a quick reminder, Minnow is a piece of software used to operate all our devices. It performs the base calling, including the calling of modified bases, along with real-time quality control and primary analysis, such as alignment and demultiplexing. It also generates summary reports from each sequencing run. Complementing Minnow, Epitome is a separate desktop application dedicated to Oxford Nanopore data analysis. The Epitome platform provides easy-to-use, pre-configured data analysis workflows across a wide range of applications for any level of bioinformatic experience. These workflows can be run locally or in the cloud, using the intuitive graphical interface. More experienced users can also choose to run them from the command line. One of these workflows focuses on the analysis of human genome variation, and this is the one we will explore in the next slides. So let's look at how the sequencing data is processed for human variation calling. In Minnow, live canonical base calling and methylation calling is performed using the built-in software Dorado. All you have to do is make sure that you've switched on modified base calling when you set up your sequencing run, so that it can capture 5MC and 5HMC modifications in CpG contexts. Furthermore, alignment to the human reference genome can also be performed in real time. This first phase of the analysis generates BAM files, which are required by Epitome. The secondary analysis runs on a computing cluster using the human variation workflow through the Epitome interface or command line. If the alignment hasn't been performed in Minnow already, Epitome analysis includes the alignment to the reference, followed by the calling of variants and methylation sites and phasing into haplotypes. The final outputs, VCF files, bed metal data, and HTML reports, support downstream interpretation. Looking at the human variation workflow in more detail, it integrates a suite of best practice recommended tools to generate industry standard file formats compatible with tertiary analyses. For instance, we use Claire3 for calling SNPs and indels, ModKit for aggregation of methylated patterns, and WhatsHap for phasing. Sniffles2, Spectra, and Straggler are used to detect structural variants, CNVs, or STRs, respectively. The workflow is fully modular, allowing our users to select which parts they're interested in, or running the full comprehensive analysis. Each part of the workflow generates HTML reports for easy visualization and summarization of the results. Furthermore, the resulting VCF files can be further passed through tertiary analysis tools to simplify the interpretation of detected variants. Therefore, Minnow and Epitome can be used together for streamlined, comprehensive data analysis of human genomes. Beginning with base calling and calling of modified bases in Minnow, through the Epitome workflow, calling all the different types of variants as well as methylation with phasing, all the way to generating final results. The output files can be visualized directly within Epitome or in preferred genome browsers, and interpreted further to address the research questions. And here it is. This brings us to the end of the workflow. So let's summarize and finish with further resources. To summarize, the end-to-end workflow from Oxford Nanopore is particularly well suited to the analysis of human genome. We have validated sample and library prep protocols that generate high-quality DNA that can subsequently be sequenced on a PromethION device to generate the required amount of data. Finally, the human variation Epitome workflow is available for the analysis of the data in a comprehensive manner. Here are some online resources where you can find out more. Take a look by scanning the QR codes. The end-to-end human variation sequencing protocol we focus on today can be found in the documentation section of our website, along with human variation protocols starting from other sample types, and further protocols covering a wide range of other applications. To view white papers, testimonials, videos, case studies, and more on the topic of human genomics, take a look at our resource center. And for more information on human genomics and multiomic sequencing with Oxford Nanopore, visit our application pages. Thank you for joining me today. I hope you found this masterclass useful.